Good morning, everybody. Today we'll talk about the first lecture in session 2 about Jalview, and in this lecture will talk about editing and coloring multiple sequence alignment using Jalview. Simply, Jalview in this objective learning of this session will talk about how to import sequence into sequence alignment into Jalview, how to decorate sequences with reference annotation, and how to build and shed parts of the Jalview, import trees, and many good stuff. You will take a look at what we are going to do. Simply by the end of these lectures, we will know how to fetch databases references for your sequences, set feature setting, change the view, using Jalview to erase multiple sequence alignment, reducing and removing gaps, and many other useful features in Jalview. When you open Jalview, you have example project. Just when you start up Jalview, you can see this example project with multiple sequences, and you can see some feature that is included in Jalview, something like how to build average distance tree, how to see 3D structure. So this the example project demonstrate key feature of Jalview. Of course, you can in Jalview you can make alignment, annotation, structure, trees, many good things for using this software. Of course, Jalview itself have many the ability to go online and fetch databases, so you can extract sequences from different databases. So you can just click on the web service and just go and fetch DB references, and then you can choose what database you would like to fetch references from it, so for your sequences. So that's a very useful feature. So whenever you have a sequence and you would like to fetch some sequences, you can go ahead and do to fetch from database, and you can get whatever references you want for your sequence and take a look at it. You can just, when you go over any of the sequences, you can see information and references for your sequences. Of course, if this you don't like to see, you can just click show annotation, or you can uncheck it if you don't like to see it and it's bother you, so you can just check it or uncheck it depending on what you like to do. Another good thing in Jalview is sequence feature, and we'll talk about it just in a second. So you can, if you go to this menu view and go to feature setting, you simply can click what you are looking for and give it a color, so you can have a colorful image of your sequence alignment, and you can give every feature in your protein alignment a color. For example, signal peptide, you can choose this color, chain, glycosylation site, disulfide bond, splice variant, helix strength, whatever. So by all that, you simply, what you do is you can do multiple sequence alignment in Jalview, you can color it, and you can use different color scheme for your different feature in your sequences. So you can have a very nice picture to include it in your publication. If you like to remove any color, you can just uncheck and it will go. Of course, Jalview default, you can see very wide alignment. So if you would like to wrap, you can click format and wrap to have all the alignment in one image, in one square image. So you can just simply click wrap and you will see something like this. Of course, you can do this if you have your sequence DNA or of course protein. So in the view, you can also, as you said, you can see annotation or not show annotation. If you would like to see sequence ID in the sequence ID tooltip, you can show it or not show it. So this is also another feature in the view. Okay, so simply why we use Jalview? Jalview is in multiple sequence alignment. It's and of course editing that multiple sequence alignment is a little bit difficult for biologists. So you need to have a program that's specialized in this. And if you do this in Microsoft Word, for example, everything will be spoiled and ruined. So one of the good feature, of course, you can also reorder your sequence, your sequence in the alignment. Simply you can press in whatever sequence you want to move up or down, and just press up or down arrow key, and then you will move your sequences. Of course, you can select a group of sequences, and all this you will see in next lectures, in the coming lectures. Simply I want you to in your own time to take a look at this domain, this protein, for example, with this number, and just bring it back and try to move up and down using the arrow key, and see how you can move from one sequence to another sequence, and vice versa. In the color scheme, you usually prefer cluster X color scheme, and this is of course Jalview give you a lot of option for color schemes. You can use whatever you want, as we will see later on. You can also hide some region that you don't like to see. So for example, if you have extremities of your protein you don't want to see, you would like to show only the domain of the protein, so you can select by the mouse, you can select the extremities and you just hide it, or vice versa, or you want to show the extremities and you don't want to see to show the core of the protein. So select the part of the protein sequences that you don't like to show, and you just click hide, and as we show you later on. You can just right click, and of course if you want to show them again, you just can click reveal, like here. You can just say, you can just select whatever sequence you want and right click and just say hide sequences. So when you do this, you simply hide this part which is selected in red, and simply you can find here a small blue rectangular. This small blue rectangular, if you right click on that and you just say reveal, you simply will reveal the hidden sequences. You can do the same thing when with columns. So in some time you would like to hide columns or hide rows, you can give you this possibility to have a better presentation of your sequence. Of course, in the view, you can do this from the view menu. You can just say select whatever you want, and then you can say view hide all but selected region. This means that you select a region and you would like to hide everything except what you select. So in this case, you will select a part that you would like to show, for example, a core of a protein or active site or catalytic site, and you simply can open view and go here and then just say all but selected region. This means that all the sequences will be hidden except the selected sequences. If you would like to reveal again, simply click in the blue rectangular and say reveal sequences. Representing a group of with single sequence with a single sequence. So simply you can select a group, for example a domain or active site or whatever, and you just select it by the mouse, and you can take it as a group. So this selection, you can say edit group, and in the box or in the text, and just edit the name description of this group. So you can select a group of sequences and put a name for that group of sequences. You can call it domain, active site, catalytic site, whatever. Removing gaps, also you can do it in Jalview. It's very easy, and every version have enhancement. So you simply can select, you can cut, you can make a selection by the mouse, and then this selection can be edited using, you can cut, copy, or do whatever you want to do in your this sequence, depending on what you are looking for. The good thing, it's as we say, that it's different from, you cannot do this in Microsoft Word, you cannot do this in simple Notepad or whatever, but it's much better to do it in this alignment editor, in Jalview as alignment editor. Other useful features, you can calculate, as we said in the first lecture, you can calculate auto consensus. So the consensus, if you would like to see it, you can also see logos. So simply you can press calculate and then auto calculate consensus. You will get the consensus of this Jalview. Of course, if you want to remove redundancy, as we mentioned before, you can simply open edit and remove redundancy. Make sure that no pair of sequences is more than X similar. So this means that any redundant sequences will be removed. As we will see in the next module for phylogenetics, of course you will need to make a tree, phylogenetic tree. Jalview will help you to do this also. You can just open calculate window, and then menu, and then you just press calculate tree, and you can choose what kind of algorithm you would like to use. Do you would like to use neighborhood joining, PFAM, average distance using PFAM? So all this kind of different algorithms, you can build a tree with it when using Jalview. Of course, we will not talk about building tree in this, but you can use this feature in the next module. This is a very small practical. You can take a look at it and practice in your own time about how to hide and reveal some regions from your multiple sequence alignment. Take a look at it and listen to the other lectures to know more about Jalview and its interesting feature. Thank you.